iteration of WISE. I think this is probably my 7th time presenting to the WISE population, which is really cool. I love the program, and it's really an honor to be invited back every year. And I'm happy to say every year I have something different to tell people. So even if you're a 4-year WISE participant, you won't always hear the same talk from me. So, but that would be terrific. Okay. So let me get going here. As Maria said, I'm working at one of the U of O Zebrafish research labs, and that is a longstanding lab at the University of Oregon. Monty Westerfield is the principal investigator there. And we use zebrafish as a model organism to study human genetic diseases. So some of those terms you may have heard, maybe not all together in the same sentence, and I'll break that down here in a little bit. Basically, we want to use the zebrafish as a proxy for understanding human health. We are not interested in zebrafish the way that a wildlife biologist or an aquaculturist or a pet store person would be interested in them. We're interested in using them to learn about us, basically. So a lot of people at this point will ask, why zebrafish? What is so beneficial about them in particular, as opposed to other things? So hopefully most of the people on this call are understand what a model organism is, and it's basically just using something else to be able to draw some comparisons or learn things about humans that we can't easily or accessibly learn in humans. And there's a whole range of different types of animal models that are selected based on the parameters of whatever the research question is that you're asking. So obviously the thing that is most like human is a non-human primate that I have at one end of this little spectrum that I made. And especially when you're studying a genetic disease, there's an advantage to studying something that's got a high degree of genetic identity to humans. However, primates are super expensive to house and maintain. They have a really slow generation time. And when you're studying genetics, you like to be able to study it through generations, parent to offspring transmission and things like that. And there's a lot of ethical controversy about using non-human primates in research at all. So there are some definite pros and cons there. At the other end of the spectrum, you have things like flies and worms. These are both great model organisms for studying a lot of basic cell biology, but also have, enough conservation or enough similarity with the way that our human genetics works that you can look at disease genes and learn things about them in there. But there are limits because of the dissimilarity, because of how divergent they are from human evolution and development. On the other hand, they grow like gangbusters. You can make thousands and thousands of experimental subjects. People don't really have any compunctions about how many you dispatch at the end of a particular experiment. Extremely fast generation time, et cetera. So those are kind of the limits of the spectrum. And I probably, with some bias, have put zebrafish sort of smack in the middle at the fulcrum here, because they really incorporate the best of both worlds, where they are a vertebrate organism like us. They have a lot of similar systems, and the development of their organs and the placement of their organs within the body is really similar. They have genetic identity that's similar enough to ours that we can understand a lot about the human diseases, which is what our lab in particular is interested in. So if you did a comparison of, how many zebrafish genes are similar to human genes in general, it's around 75%. So if 75% of human genes, you can find an analogous version of that gene in the zebrafish genome. But if you restrict that question to how many of the human genes that are linked to disease can you find represented in the zebrafish? That number goes up to 85%. So those are pretty good odds in order to be able to find the thing that you're looking for, and then ask some specific research questions about what is the molecular basis of this particular disease. So one of the really wonderful, beautiful things about zebrafish, no matter what you're studying, disease or otherwise, is how lovely their development is to look at. So we have they're very small relative to, like a mouse, or certainly a rabbit, or some other mammalian organism. We can keep 20 of them in a 4-gallon tank, that's filtered water and everything like that. So the footprint of keeping a lot of different research stocks is pretty small and less expensive. And a male and female mating pair will create thousands of eggs that will then, be fertilized and developed into young fish. And the development is really rapid. So you can see the abbreviation here, HPF, that means hours post fertilization. So there's a 1-cell stage, 15 minutes after sperm enters egg, and then less than a day later you have something that already looks like a fish. And that's fast. You get, if for so comparing to human development, for example, this takes about 3 weeks to get to something that is at this stage. And as you can notice, there's nothing that's been done to these embryos in order to photograph them, except for to remove the transparent egg case that's around them. That's called the chorion. So the tissue itself is completely transparent. You have these, this little ball of dividing cells that's sitting on top of this sort of golden globe, which is the yolk sac. And as the organism grows and develops around it, it starts to consume as soon as it gets something like a gut, it starts to consume the nutrients within that yolk sac. But everything else is completely transparent. So you can see the organs and systems developing in real time just with a high quality light microscope. You don't need to do super fancy special things for a lot of that. And then at about 3 days, they hatch out of this chorion and start swimming around. At this point, they have this is a 2-day animal that's been removed from the chorion. You can see a heart. If this were a live video, you would be able to see the heartbeat here. The eye has pigment. You can see sort of the chambers and sections of the brain up here, if you know what you're looking for. Here is the ear. There are muscles and nerves. It can swim, it can respond to the environment. And then by 5 days post-fertilization, or 120 hours, it's eating, it's pooping, it's swimming, it's singing, it's hearing, it's responding. So you have this whole array of behavioral of characteristics that you can evaluate, as well as how it looks and how its organs formed and things like that. So another advantage to it being transparent is that you can make transgenic modifications to make particular proteins or structures glow under fluorescent light. And I just have a couple of them here just because they're so pretty. This is a muscle protein that's labeled here, and you can see it mostly in the muscles of the body, but then there's a little bit in the heart too, because the heart has important muscles. This is a protein that's present in all of the blood vessels, so you can see big action here at the heart, but also the eye, the brain, all through the body, this large blood vessel that's carrying blood to and from the tail and back up. And in real time, you can just sort of see them swimming around and then watch the development of these blood vessels. And if there's a problem, then you can see it happening. You can see it unfolding in real time. And then this one is really cool. There's 3 different trans genes in here that are glowing with different fluorescent wavelengths that are lighting up different populations of neurons in the forebrain. These are photoreceptor cells in the retina. Here's a midbrain, and there's some other ganglion cells here in the retina that correspond as well, and then in the hindbrain, and then yet there's a 3rd population in the retina too. So different neuronal markers that are showing you different things about the development of this 5-day-old zebrafish nervous system. So how about that?
Uh, Dr.
um,
Phillips, it looks like
yes.
we have a question in the chat.
Okay.
Elliot, which part of their body develops first?
Um, well, I mean, like a lot of vertebrate organisms and even invertebrates, a lot of times it's the nervous system that really starts building. I think, the nervous systems, even in the like invertebrate models, nervous systems, I would argue, are one of the most complicated systems, and take a lot of kind of layered wiring and construction. So to me, it kind of makes intuitive sense that they would they would take longer to build. So you had to get an early start on that. And it's also kind of a make or break thing. If you're an organism developing and something goes wrong with the nervous system, maybe just stop and don't do all of the rest of it, because that's a deal breaker. If you can't get a functional nervous system going, then the rest of it doesn't really matter. So yeah, that's you can start seeing brain and spinal cord before a lot, and even before it starts to look like that, the cells that are going to specifically become that are differentiating the cells very early on in the process. Okay, so you don't even need a fluorescent microscope to be able to see stuff inside the fish. This is a chemical stain that we use pretty routinely to look at cartilage and developing bone in these young fish. And so the blue is a chemical called alcyon blue that just binds to the sugars that are in cartilage and shows us where they are. And then these young guys, most of it is blue. They're still pretty soft and gushy, and they're only just beginning to develop their bones. You can see a little bit of dark red back here. There's teensy little bits in here. In the fullness of time, and indeed just a few weeks later, in this guy, you can see a lot more red. It's growing, it's grown a spinal cord at this point, a little fin rays. There's still plenty of blue cartilage in here in the forming cranium, but there's more red appearing. And there are a lot of human diseases that involve skeletal growth and development. So being able to look at these things that are happening in real time is also really valuable research tool for us if we're looking at diseases that affect the skeleton. So using a model organism for this purpose, not just to learn things about life because it's cool, although that is definitely a true thing. It's more of an opportunity to bridge that basic science inquiry with something that you can apply to human medicine. There are a lot of different human genetic disorders. Some of them are extremely common, some of them are quite rare. But if you lump them all together, 1 in every 200 babies born is either has something or is going to develop something due to a faulty gene that will affect his or her development or life or anything like that. So it's definitely a worthy thing to be asking about. So one of the important things, in addition to just understanding the molecular basis of these various genetic diseases, is that if you're going to actually approach it from a point of what can we do for people who have this particular disease, what can we, how can we treat it, how can we prevent it, how can we screen for it, any of those things, it's really pretty much a requirement to have some sort of a model organism to ask those questions in before you get to being able to apply it to humans. So that's another good reason to develop these models and study them, because it's another bridge to that application of being able to do something for the people that are affected by that. So I don't know how much you have talked about genetics in the 10th grade biology class. I'm sorry I wasn't able to find this sooner. Maybe Mr. The instructor can give me like a thumbs up or a thumbs down to know that if we have a basic concept that if you have a genetic disease, it's because you have something that's faulty in the code of your DNA. And then that by virtue of translation and transcription in the cells is going to usually manifest as something that's inoperable or suboptimal about a resulting protein that's made. Is that's
Right. We've
not news?
definitely talked about transcription and translation
Okay,
and
good, good.
mutations.
That's good. Okay. So I just want to make sure we're all on the same page with the basis of that. And so one of the disease genes that I've worked on for a few years that we've done a lot of pretty quality informative research on is a disease called Usher type 1F. So Usher syndrome is a hereditary deaf blindness. There are a lot of different genes that when mutated will cause this disorder. Um, and some of them are extremely common ways to get Usher syndrome, and other ones are quite rare. This is one of the ones that's globally quite rare, but it's because a lot of genetic diseases and the frequency of them depends on, how isolated a particular population is, or, who married who generations ago, and that sort of thing. This one is extremely enriched, you would say, or the prevalence is elevated in the population that's descendant from the Ashkenazi Jews. So overall, all types of Usher syndrome in general it's a frequency of maybe 1 in 10,000 people in the whole world um, have Usher syndrome, and quite a few more of those are carriers. But in the Ashkenazi Jewish population, you have a carrier rate of 1 in 50. So that's significantly higher. This is all types of Usher syndrome, including type 1F, are recessive disorders. So two parents who are carriers have a 1 in 4 chance of having an affected child. And again, within this population, this is the leading cause of hereditary deafblindness, for sure. And because of that, that isolated population phenomenon is what we call the founder effect. Most of the people in this population who have Usher type 1F have it because of one particular allele, one particular specific genetic change that both of the parents had inherited and passed on. And that is a truncating, it's a point mutation that converts an arginine codon to a stop codon instantly. So instead of this very long multi-domain protein that does a lot of important stuff, you get a stumpy protein that is, relatively non-functional. It can't go anywhere. It's going to just kind of clog up the works and not be able to participate in any of the things that the normal, or as we call it, wild type protein would be capable of doing. And so just to remind everybody that while we talk very technically about human diseases and disease genes and things like that, there are always the people behind these stories who are living with these disorders. This is my friend Jessica that I just wanted to introduce you to, who she and her sister both have Usher type 1F. And so this is a hereditary deaf blindness, and Usher type 1, it is a severe form of it. So they're born with profound hearing loss and balance problems. They can't learn to speak unless they have a cochlear implant. So Jessica and her sister both got cochlear implants when they were toddlers, and they're now adult women. Jessica's currently in graduate school, and she wants to be able to be a sociologist and do research on disability access, because she's learned a lot about that in her education as somebody with a hearing and a vision deficit. But she has concerns about, not being able to do those things if she has diminished vision along the way. So it's a progressive vision loss that's happening to her over decades, and it's getting worse. So the highlight of that is that there's time to do something. There's time for an intervention of some sort of therapy if we could just figure out what to do. So that gives our work in the lab with the zebrafish a lot more kind of impact and a little bit of urgency as well, because for all of these people, for Jessica and her sister and the thousands of other people who have Usher syndrome and Usher type 1F in particular, the clock is ticking. So, so we have motivation to get going on that. So let me give you a real brief introduction to the cell types that we're looking at that are common between the humans and the zebrafish. So in our inner ear, inside the cochlea, in the case of humans, we have thousands of little cells that have these specialized projections that come up out of them. These are all kind of tethered together. And so they move in concert when they're impacted by a sound wave or some sort of emotion sensation. And when they move, they open up channels within the cell that they're attached to. And that creates an electrical signal that ultimately goes to the brain and say, hey, you just heard your mother calling you for dinner or something like that. So all of the organization of this structure is put together by Usher proteins. This one that's in blue here, protocadherin 15, and they're so tiny, you can't really illustrate them on here. It's a little bit clearer here, but still protocadherin 15 here. That's the protein that is involved in Usher type 1F. Cadherin 23 that it's tethering to here and holding these two sides of the larger structure together is the protein that is affected in Usher type 1D. This one's Usher type 1C. This one's Usher type 1B, etc. So that's why there's a hearing defect and a balance defect in people with Usher type 1. It's because this whole assembly holding this thing together is broken when you lose one out of that. It's like an essential game of Jenga where one thing comes out and the whole rest of it falls apart. So that's what's happening in the ear and in the balance organ of both zebrafish and humans that have this impairment. So our question was, can we create a mutation in the zebrafish that's going to emulate this particular Usher 1F mutation, the R245X, to the point that we can, excuse me, that we can learn something specific and actionable about it. So, step one was let's test and see if it actually has the same symptoms. Here are the zebrafish structure that we were just talking about. These are pictures that I took of our fish after we made this. We used CRISPR-Cas9. We targeted a part of the exon that contains the corresponding codon that's affected in the human patients here. We made a premature termination codon right in the spot where we wanted it to. So the resulting protein is going to be truncated in the same way as it is in the human patients. And indeed, it's broken. We can see the splayed bundles that are supposed to be nicely put together in this process when we look at the fish that are affected by this mutation. And then I can show you what they look like. Ooh, that's loud. I'm going to turn that down. So this is a really easy test we can use on the zebrafish to see how well they can hear. You tap on the dish with this is basically Sharpie, whatever, something that's gonna make a startling tapping sound. And the fish have this very prescribed way of kind of darting forward in a straight line to evade whatever the thing is that's making that noise. And it's a really programmed response. If you can just watch and see them all, kind of leaping forward in a really similar dynamic kind of way. In the Usher fish, I'm sorry, I should have turned these volumes down. Most of them aren't moving at all. They're not hearing that tap response. And the ones that are doing this little loopy swimming pattern. Can you see those guys like looping around? So if they could hear it, it means they want to startle, they want to do that, but they have such poor balance that they are just swimming in a circle. So, and that's a really typical swimming pattern of an Usher impaired zebrafish. And it's indicative of the balance problems that are caused by having those genes defective. Let me go. Oh gosh. Okay. And
Really quick, we've got a few questions in the chat.
okay,
Give
sure.
it a second. So here's a question that says, apologies if this is a morbid question, but what is the most interesting genetic disease in your opinion?
Hmm. You know, I um I'm not sure that I have one discrete answer to that. I mean, they're all interesting. And that's, sort of the balance of being me is that I can really, really geek out about the biology of what's underlying this. Um, but also keep in mind that there are affected people and it's a very serious, tragic thing. So I feel like Usher syndrome, I've been studying it for a long time and it is really interesting to me because of the fact that there are these disparate cells in the eye and the ear that are basically using the same molecular toolkit to do their thing, and and that's neat. I mean, photoreceptors in the eye, which is what I was going to introduce next, and I don't have to if we are done talking about that. Are doing a very specific environmental reception and transmission thing, and the hair cells in the ear that I just talked to you about are doing a sort of parallel environmental to neurological conversion of that environmental, that sensory information. So the fact that they're using the same molecules to accomplish that but in really different ways at the subcellular level, I think is quite fascinating from a molecular biology standpoint, from an evolutionary biology standpoint. The diseases that are really sort of medical mysteries, I think are interesting from a standpoint of, I mean, that's part of our role. And I can skip ahead to that if you want um, to talk about that more, because yeah. So here's another element of what we're doing that has nothing to do with Usher syndrome. We work with a group of clinicians um, and geneticists all over the country who are seeing these incredibly rare diseases that don't have names yet. They don't have a genetic diagnosis. They don't have a way of saying, maybe this is like the first patient that this particular doctor has ever seen with this set of symptoms in their entire career, and they don't know what to do with them. So there's now a sort of an umbrella organization called the Undiagnosed Diseases Network that we're kind of put all of these, the doctors, the geneticists, the model organism screener researchers like me together and say, okay, well, let's figure out what are the most likely candidate genetic causes of this, and then let's figure that out using the zebrafish, or in some cases the fly or the worm or the mouse, to validate the fact that this is really the disease that's causing this problem. And along the way, a lot of the doctors who thought they had the only person in the world like this will meet like 6 or 7 other, doctors, and then you have a little group that has these, common denominators and similarities. So it works really well. This is just an example of that, where all of these individuals have a very specific kind of facial development presentation in the clinic, and they also had some vertebral abnormalities. I know I'm not going to go through like what exactly they are, but the paper describing it is that there's some abnormal ways that the bones are growing and forming. And additionally, a problem with their white blood cells, which are the infection fighting cells in our blood system. And all of these patients in the study were found to have at least one mutation in a gene that hadn't been linked to human diseases before, called tonsil. It has nothing to do with your tonsils. This is just the naming convention of proteins. TONSL stands for something really complicated. I'm not going to say it. I'm just going to call it tonsil, but just don't, the tonsils in these cases are completely unaffected as far as I know. So we created a similar mutation in the zebrafish. We found immediately that there was a big problem in the way that these zebrafish grew and developed. They had a general symptom that we kind of call failure to thrive, and that they were eating food. That's what that little arrow is pointing to, is like the food that they're swimming around and eating, but they look just shaggy and terrible, and we're really having a rough time. Um, and eventually they didn't survive much past this point. So they had a real absorption problem with nutrients. When we looked at the neutrophils, those white blood cell components in their gut, we can see a real big depletion. So these are normal. This is a normal load of those neutrophils in the gut that I outlined here. And then there's a big range of how full the gut is of neutrophils, but the ones that look the healthiest have the most. The ones that look the unhealthiest have the least in this situation. That kind of tracks onto this. And they also have some problems with their vertebra. These little pink, this is that bone stain that I showed you again. So the little pink things in a normal fish are the first part of a vertebra that's forming. This, they're gonna make these little pink rings around the cartilage that's there in the backbone, and then actually ossify it, just make it into bone. So that is happening at a very accelerated rate here in the mutant zebrafish, which has some correlation to what went wrong in the human individuals that were affected with this disease. So we use the zebrafish exactly as designed by this Undiagnosed Diseases Network to be able to make a copy of what we could see and tie all of those, the patients that were coming from all different corners of the United States, to be able to say this is definitely the gene. And then we published that, and it is indeed definitely the gene. So now that goes into the database of human genetic diseases, and the next time a doctor has somebody like that come into their clinic, then they'll be able to validate it a lot more readily because of the work that we did. So so that I feel is interesting. And I have, we have with the Undiagnosed Diseases Network, we've done at this point a couple of dozen of disease genes that have been passed to us by the human geneticists and the clinicians. And we have several more papers that have those types of findings to be able to put a name on a thing that previously was an unknown entity. So, and just real quick, and I'll tell you one more thing about what we're doing with the Usher 1F fish right now, who clearly have all those symptoms, is we're starting to test small molecules, like sort of proto-pharmaceutical compounds, to see if we can make a difference to their symptoms in any way. So this is one of the ways in which we're hoping to be able to help people like Jessica, by if we can find the right molecular compound to put on the little fish when they're developing, and change the course of the disease in some way. Maybe their retinal cells look healthier at the end of the testing period than they do in an untreated fish. Maybe their balance issues get a little bit better or something like that. But that's one of the things that's super straightforward and convenient to do with zebrafish, which I'm sure I don't have to explain why this would be a lot more challenging in a mammalian system. But it still gives you a whole organism to look at the effects of. So we're looking at the eyes and ears for specific effects in this Usher syndrome case, but we're also making sure that the drug we're putting them on isn't giving them any kind of like liver toxicity or other developmental problems, because we certainly wouldn't want to promote something as saying, yeah, take this, it'll cure your vision, but it's also going to make you infertile, or something like that. Is kind of the other half of any kind of drug testing. And again, we would absolutely need to do that in the zebrafish before we could sort of pass it up the chain to something that would approach a clinical trial. So I'm going to shut up and hope that you guys have some more questions. Maybe I should stop sharing my screen and I'll just talk with my hands.
That'd be great.
Okay.
Yeah, so we've got a few questions in the chat, and just a reminder, if anybody has any questions, send them in the chat and we will read them out and get them answered for you. So here's a question This person is saying, I have two questions. The first is, do fish get different diseases from humans? And if so, what are the complications you get from that when working with them?
So that's a really good question. So the diseases that we work on are genetic diseases specifically, where the gene that is underlying the symptoms of that disease has to be mutated or affected in some way. Obviously, there are a lot of other ways to get disease, including, bacterial or viral transmission and things like that. And occasionally there is a little bit of a risk from that things can communicate from zebrafish to humans or vice versa. It's pretty low in terms of things that infect us, like other organisms. Our bodies are different enough that we don't have a whole lot of common pathogens in that sense. But there could be a person who would be immunocompromised, for example, would have to be more careful in working around the fish water. There's just like in our environments, we have a whole lot of, benevolent or kind of neutral bacteria and other things that are not pathogenic to us if we have normal, healthy immune systems. But when we do have immune compromise, maybe some of the bacteria that wouldn't affect a healthy person might have a bad impact. It would be the same thing with the zebrafish environment, because there certainly are, bacteria and things that live in there. There's a few really bad known ones that we have surveillance for all the time, just to make sure they're not in our water system. And we know to look for them when we know how to treat them. So it's never been a huge issue, but we also have a lot of security protocols to prevent that from happening. There's real restrictions about who can go into the fish colony room, and you have to wear gloves, you have to wear shoe covers, you have to clean your hands with ethanol when you leave. We were doing that way before COVID. Yeah, it's to protect the fish from us and to protect us from the fish in the very unlikely event that something like that would happen.
Cool, thank you. One other question. When you're looking at the amount of time a fish might live after getting a disease, how does that compare to a human with the same sickness?
That's a great question too. So it really depends on how survivable that particular thing is. I mean, on a scale of, a human lifespan, say, all things being normal and equal, 78 years or something like that, a zebrafish lifespan like that is around 2 to 3 years. So everything is scaled pretty short in that way. And then if it's something that causes a disease, we're really looking for a correlation between at what time of the individual's life does that disease manifest. And that often we see is very similar in the juvenile fish versus the teenage human or things like that. And then it depends on how survivable it is. If that thing happens to the fish, is the fish going to be able to swim? Is it going to be able to eat? There are a lot of modifications that you may be able to make for a human individual with that disease that you can't make for the fish, and then this fish isn't going to survive it. So hopefully that answers the question.
Great, thank you so much. Any other questions? Put them in the chat.
So thank you, Dr. Phillips, for your time and for your great presentation. And Dr. Phillips, and thanks for all the times that you've come and presented for the WISE Symposium.